Thank you very much. I am not going to continue in Hebrew. I hope that's okay. So what in this opening talk about, I think we're going to try to talk about is what does it really take to deliver high quality diagnostic results, and in particular for hereditary conditions, using the brand new technologies of next generation sequencing. And then you'll hear a little more from Drs. Powell and Friedman about the clinical applications of that, although I'll touch on it as well. I'm an employee of Invitae, and I speak only for Invitae, of course. And what we're trying to do at Invitae is to essentially address the fact that genetics in clinical practice tends to be rationed, right? Many patients could benefit from genetic information, and many more will in the future, with additional genetic information becoming available about the impact of certain genes. Then today, by the way the system works, folks will get that testing and have it involved, in decision making in their healthcare. And we think this is going to be important in the end for everyone who at least is under a modern medical system everywhere in the world. So as a step towards that, just one step, we have developed a set of gene panels for testing for hereditary conditions. A significant focus of ours, and what we'll be talking about mostly tonight, is hereditary cancer, where we have 29 genes, all of which are known to be clinically significant to various degrees. We also have fairly comprehensive offerings now in a couple of conditions within cardiology as well as neurology. And again, all of this information is available on our website and is available from the folks at Teva. And why do we take a panel approach? Well, again, I think you're going to hear more clinical stories from Drs. Powell and Friedman shortly. But just to summarize, by testing for more genes up front, we know that we can get a higher diagnostic yield, and I'll show you examples of that. We can shorten time to diagnosis. We can eliminate some of the sequential testing cycles that can be both very expensive, laborious, and challenging for the patients. And we can address cases where the presentation, either within the proband or the family, doesn't necessarily cleanly fit what's been well documented to date in the clinical literature. We know that many of these genes are pleiotropic, and we know that the spectrum of phenotypes can vary for the carriers of these genes. We were involved in one of the first papers, as have some of the others here, on the use of these broad hereditary cancer panels. This is just a reference to our April paper in the Journal of Clinical Oncology. Again, I think you'll see many other interesting references on this topic if you're want to read up on the background. Some of the challenging things to do to make this sort of panel testing broadly accessible include not just getting the science right, getting the technology right, getting the clinical utility right, but many other things around trying to make reports easy to understand. Most clinicians who are treating cancer patients are not specialists in genetics. And so we've done what we can, as have others, to try to make the information maximally useful, still complete, but at the same time having the genetic counselors and the clinicians not have to wade through lots and lots of detail, particularly if they're not specialists in genetics, and they're looking essentially for the bottom line answer to make the next decision they have to make with the patient. Another thing that we've also done is try to address the fact that today, capturing family histories, which of course is vital when you have patients who may have hereditary conditions, tends to be a little bit difficult and time consuming. So we've developed a family history tool, which is now available on the internet, on our website, and as well on the Apple Store for iPads. This is free to use, so everyone in this room is more than welcome to use this. You do not just have to use it with Invitae tests, and you may find it a convenient way to capture information from your cases where there are strong family histories and various attributes that you want to make sure are recorded and documented. And there are sharing features, so for example, if you've referred a patient to another clinician, you can share the pedigree with them. And that's all done securely. So I'm going to now take a few minutes and talk a little bit about technology. Now, I'm imagining most people here are not specialists in lab technology. So I'll apologize up front if I use a few words that may be unfamiliar to you, but I'll try and hope everyone here understands the basic points. And the key observation is this. This is what a DNA sequencing lab, a fairly large one, looked like even just 10 years ago. Rows and rows of these machines called capillary electrophoresis machines, and for the older people here, we remember slab gel machines, which were even more ancient. And indeed, a building full of these machines, right, or actually 5 or 6 buildingfuls of these machines, were required to sequence the first whole human genome, which of course was done at the cost of approximately $3 billion US and took about 13 years. And that's why we had diagnostic tests offered clinically that use that same technology that cost typically at least $1,000 US per gene and up from there. What has been invented and has emerged over the last roughly 8 years has been something called next generation sequencing, a term that many of you have at least heard, and certainly you will be hearing more of. Those machines look like this. And even 1 or 2 of these boxes can sequence, at least to research grade quality, an entire human genome in a day for about $5,000 or less. Conversely, you can run dozens or hundreds of clinical grade, very high accuracy diagnostic panels on that same machine in 1 day. And this has led to a revolution in how we think about the use of genetic information. If for no other reason, then we're able to remove a lot of the time barriers and cost barriers to generating that information for your patients. But next generation sequencing is not without its challenges. There's people from the companies that sell this equipment and sell the reagent kits who will come to you and will say, oh, all you need to do is buy our box and buy these reagents and download some software and you'll be fine. And I wish it worked that way. It doesn't work that way. Indeed, there's a number of interesting technical problems that I won't go into all the details of, but these NGS reads, they are voluminous. Data sets coming off this machine can be terabytes in size. And one needs very sophisticated bioinformatics algorithms and methodologies, both to quality control that data and indeed to make sense out of it. For the reasons that I mentioned here, these fragments that come off the machine are messy, noisy, small, and you have no idea where they came from. You have to piece them back together into a picture of what's going on in your patient as if it was a jigsaw puzzle. There are solutions, and again, I won't go through all the details, but I'm happy to take questions on it. But effectively implementing diagnostic grade whole sequencing of genes, gene panels, and indeed exomes or genomes is something that can be done, but there's a very substantial difference in the quality and completeness one will get if you've invested heavily, if your laboratory's invested heavily in the bioinformatics, in the biochemical research and development to address a number of the technical limitations of these machines. And they do have limitations, but again, the benefits, if you can solve these problems, are that they generate very, very useful data. They do it inexpensively and they do it quickly. Just to give you one vignette of that, in our laboratory, we want to make sure that we are able to assess comprehensively the different types of variation that lead to human disease, a spectrum that in previous years would require multiple different technologies perhaps to be run on any one patient in order to generate an integrated report. From the next-gen data alone, we're able to apply multiple different computer algorithms, each of which pulls out certain types of variation, merge that data together into a comprehensive view of what's going on in the patient. Again, unfortunately, you can't just go download the software package and run it. It requires quite a bit of tuning and maintenance and investment in the R&D and the bioinformatics and IT infrastructure to run it. But properly done, we can generate some very thorough views of what's going on in a patient, again, from a single laboratory technology. I'll show you an example of that in a bit. But let me just level set one thing. Has anybody here ever seen a picture like this? IGV plots? I didn't think so. This is the classic way people look at next-gen data, and you don't have to understand all the details of this. But suffice it to say that one of the attributes of NextGen that's both a blessing and a curse is the fact that it will generate reads, these individual gray bars, that come from each of the exons that you're trying to report on for your patient, but it does so very unevenly. So for example, in this particular gene, these two exons wind up being very deeply covered. Over and over and over and over again, the machine is generating raw data. And it's that massive redundancy that helps overcome some of the other limitations of the platform. The fact that we can do this 50 times over, 100 times over, 200 times over, 500 times over. But while these exons here are very deeply covered, some other exons are not. And this leads to some interesting challenges. And you'll hear different laboratories throw out the quality metric of read depth or coverage. And they'll throw it as if, oh, well, we do 100x coverage, and that's better than this other lab that does 50x coverage. That's sort of true, but I want to leave you with one important point here. The thing you probably care about for your patients isn't the average, right? It doesn't really help you if this goes down 500 or 1,000 times. What you care about for your patient is this one. It doesn't go down enough. And so we tend to focus mostly not on the average, we focus on the minimum. We want every point that we have to report on for that patient to have at least 50 of these gray bars, 50 times oversampling, 50 times redundancy in the raw data in order to make sure that we're producing a complete and consistent and correct result for that patient. But it turns out there's an interesting observation that we and others have made, which is while exon to exon to exon, as cartooned here, this read depth can vary, right? You will get many more reads from some exons and fewer than in others. But if you look across different patients, the profiles actually tend to be very consistent. The read depth varies, but it consistently varies between patients. And that actually starts to generate data that look quite a lot like copy number microarrays. CMA analysis, where, for example, when we see a patient that fits that profile but deviates in one spot, this would be the hallmark of a duplication or an amplification in this particular patient. And again, the methods for dealing with this are not simple, but they all indeed leverage the many years of experience that we and others have in running copy number microarrays. The techniques for dealing with these data statistically and from a quality control and laboratory point of view indeed are very similar to what's been long published in that field. But there's something you can do with the next-gen sequencing data that you can't do with the microarray, that you can't do with the traditional laboratory technologies. And that's this. Even when we're looking at a large deletion of, say, BRCA1 or perhaps a duplication, we're able to see it not just as a copy number change, but also as a sequence change. So for example, because this patient's mutant allele will actually have these two chunks of DNA contiguous with each other, because they're deleted, we'll actually see that contiguous chunk of DNA in a next-gen read. And that allows us to very accurately pinpoint the breakpoints of the mutation in that patient. Again, something that you can't do using the older methods. You can ask whether that really works. And here's one example here. This patient has a 126 base pair deletion of BRCA2, something that traditionally has been considered difficult for next-gen sequencing because it's a big deletion, a big indel, as we like to say. But at the same time, it's a small copy number change. So it's too big to be an indel, too small to be a copy number change. It lives in this gray zone that everybody struggles with. But again, adding this, what we call split read signature, these big colored blobs here show you exactly what is going on and indeed exactly where the breakpoints of this event are, which happens to take out most, but not all, of this exon or BRCA. We have many other cases I can show you, again, where the colored blobs, if you have one take-home lesson, colored blobs are interesting, where the colored blobs stand right out at you. What's interesting in this case is that this patient has a duplication of exons 5 to 11 that was detected, again, using traditional technology on a microarray, was detected by us using the read depth approach I mentioned. But duplications, if they're novel and uncharacterized, are difficult to know whether they're pathogenic. I can take a chunk of a gene, duplicate it somewhere else in the patient's genome, and it had no effect on them phenotypically. Or I could take that chunk of the gene, duplicate it in tandem, and it has quite a substantial effect. And indeed, because we can look at the sequence here using the power of next-gen technology, what we're able to see very quickly is indeed this is a tandem duplication. It's one that hadn't been reported previously in the literature. There is not any specific functional data on it, although just from looking at the sequence exactly where and how this chunk of DNA got duplicated in tandem and inserted into the middle of exon 11, it turns out it looks very clearly pathogenic. Now, I think we report it as a likely pathogenic because, we still don't have hard data on it. But I would, certainly suggest that this patient is likely to have a BRCA phenotype, and indeed, the clinical history of the family is consistent with that. Events like this turn out to be difficult for some of the traditional methods, but next-gen sequencing can catch. Here we have a deletion of an intron, this part of the genome in between two of the coding exons in this gene. And again, because of the next-gen technology, we can pick up that effectively what you have is a fusion mutation in the patient's genome, something that we typically expect to think about in cancer cells as a somatic mutation. This patient has one in their germline, heritably. So that's a bit of the technology. Again, probably not something that most of you have to spend too much time dealing with on a daily basis. People like me have to live, eat, sleep, and breathe it. But perhaps something that is relevant for everybody in this room is to think about how do you validate this kind of approach? How do you know that it's both technically and clinically giving you the answer that you expect to get and that you need to get at the quality you need to get it? So we've assembled a fairly large cohort to answer that question, over 1,100 samples, 1,100 samples. The single largest group of which, these 735 here, are prospectively accrued following the NCCN guidelines for hereditary breast and ovarian cancer counseling. And we were able to ask the question whether applying multi-gene panel testing to this large group of patients produced data that, first off, were consistent with the earlier traditional testing that these patients had received, in this case almost always from Myriad Genetics. And then secondly, we could ask the question of if we're now testing for more genes, what in addition are we seeing and what are the potential clinical implications of that? Well, on the first question, the answer turns out to be simple. We were 100% in agreement. And that includes some of the tricky cases that I mentioned earlier. Some of these large insertions, deletions, complicated mutations, very small deletion duplication events, copy number changes, and everything else that was present in these families. We had 100% agreement with the prior testing. But importantly, and where I think this study really went beyond some of the others that at least I've seen published in the literature, is that among the mutations turned out to be a fairly significant number of these difficult types, again, including particularly things like the single exon deletions, single exon duplications, and other complicated sorts of variants. And by complicated here, I mean not necessarily complicated to interpret clinically, but complicated to detect accurately in a laboratory. And indeed, where we know some other methods can at times struggle. Now you might say, oh, that's nice, and it's good that you have many of them, whereas some other validation experiments have few. How relevant is that clinically? Well, it turns out the answer is very relevant clinically. So let me give you a perspective on that. So we, again, are looking at a 29-gene cancer panel in 1,100 individuals. If you count all the benign variations, does anyone have a guess at how many different sequence variants there are in 29 genes on 1,100 people? That you don't. The answer turns out to be almost 60,000. There's a lot of differences between the DNA, most of which have absolutely no clinical impact, most of which are very simple single nucleotide polymorphisms, things that almost any good laboratory technology can detect very accurately. So in other words, these complicated events turn out to be less than 0.05% of the total spectrum of variation in these patients and these genes. But interestingly, if we look at the pathogenic variants, the ones that actually cause disease in these patients, they're a very disproportionate fraction of the pathogenic variants. 10%, perhaps even 15%, depending exactly how you want to count, of the pathogenic variants in these 1,100 patients are indeed these technically tricky cases, and indeed a rather small minority of the variants in this population are the simple single nucleotide changes that indeed most platforms can ace easily. So it actually is important to focus on these difficult changes if you want to make sure that you haven't missed a pathogenic variant in your patient so that you can ensure that you're giving them the best care possible. Perhaps more relevant clinically would be now if you compare the 29 gene panel results to the prior mostly BRCA1 and BRCA2 testing, what do you find that hadn't been detected previously? And I know this topic will be addressed far better than I can by Drs. Powell and Friedman, but just to give you the numbers from this study. What we find, and I'm going to focus on the dark bar on the bottom here. This is the prospective cohort that I mentioned, the one that's maximally clinically relevant. Now I should be careful to note, this is a US population, so not necessarily emblematic of what you would see in Israel. Also importantly, it's a US population from Boston and Palo Alto, which means it's about 75% Caucasian. There are definitely quite a number, I think around 85 Ashkenazi, self-reported people of Ashkenazi descent on both sides of their family. But it definitely has a underrepresentation of minorities in America. Nevertheless, for what it's worth, if you look at that prospective population here, what we see is that the diagnostic yield increases fairly significantly as you add additional genes to what you test the patients for. And I'll give you some vignettes on that earlier. In the prospective cohort, most of the additional findings tend not to be a small number of other genes, such as CDH1 and STK11, that are known to be perhaps as high penetrance, or as bad news for the patient, if you will, as BRCA1 and BRCA2. But by contrast, a significant number of the findings are in these moderate risk genes, genes like ATM and NBN, that confer twofold to threefold to fourfold relative risk, leaving many patients, depending on other factors, with lifetime risks of cancer in the 20% to, say, 40 to perhaps 50% range. And these are fairly prevalent among this population of individuals with cancer. Also prevalent has been Lynch syndrome genes. Lynch syndrome genes, which are typically thought to be associated with colon cancer and endometrial cancer, turn out also to be fairly prevalent. Again, not only among patients with ovarian cancer, and there are some in this cohort, there's a known link between Lynch syndrome and ovarian cancer that's fairly well established, but also even in the families who are only affected by breast cancer, at least at this time. Now, interestingly, we also see very few genes pop up which are not currently known to be involved in breast and ovarian cancer. And that's also reassuring. We wouldn't expect to have seen this bar be very large, particularly the dark blue one on the bottom. Which means that our clinical knowledge of the genetics of cancer may not be 100% complete, but is fairly complete for most cases. And just to give you a view of some of the genes that indeed have been seen as positive in this study, again, PALB2, CHEK2, and ATM, again, all relatively moderate risk genes, at least compared to BRCA1 and BRCA2, come up fairly frequently in this study, as does, for example, PMS2 and the other genes that are involved in Lynch syndrome. So what was the clinical outcome overall? It was interesting. So again, if 9% of the population, as expected, was BRCA positive, another 4% was positive for one of these other genes. And when we went back and looked at all the clinical records, it turns out that almost all of the time, when we were able to get good family histories and good clinical records, what we saw in the family was very consistent with what we know about that gene. These were apparently not incidental findings. They were related to the patient and their family's phenotype or both. Also, perhaps most importantly, we asked in as detailed a way as we can, using multiple different physicians, whether you would act on this information. Under current guidelines, which still don't speak directly to most of these genes, would you treat the patient differently given this information? And the answer turned out to be 70% of the time, the answer was yes. And the different things they might do ranged in spectrum. There were a handful of cases where they might consider recommending to the patient prophylactic surgery. More cases were just advanced screening, accelerated screening, screening of family members were indicated. And finally, what they found, our clinical colleagues on this study, was that in the genetic counseling process, the information was both feasible clinically and was appreciated by the patients, in spite of the fact that one is now communicating information with far greater uncertainty to those patients compared to BRCA1 and BRCA2 traditional testing. And we have many good examples, but I'm going to defer to Drs. Friedman and Powell on some of the clinical stories. So in summary, I'd like to thank all the people that I've had the pleasure to work with at Invitae, as well as our colleagues here in Israel at Teva, and also our friends at Stanford University and Mass General Hospital who provided the patients and much of the clinical insights. I think I'll stop here. We might have time for one or two very quick questions, and then we'll move on to the other presentations. Thank you.
Hi. What is your approach in Stanford towards testing for moderate risk variants for non-symptomatic for the healthy members of a family?
Right. So in all of these cases, the patients were indicated for testing under current guidelines, but I think 85% had a personal history of cancer and about 15% were indicated for testing because of an affected family member or a family member who carried a known mutation. And so this study looks specifically at the case of that, but when, based on family history at least, the guidelines for testing were met. In terms of testing completely asymptomatic individuals with no family history, this study hasn't looked at that, but there are other studies which have, and maybe Drs. Powell and Friedman can address that as well.
What is the efficiency of your system to detect gene rearrangements compared to other known commercially available platforms?
So he was asking about efficiency for detecting gene rearrangements. So gene rearrangements that are copy number changes, that induce copy number changes, again, we have found no discrepancies between the traditional testing approaches and what we've done with the next-gen sequencers. There are a couple copy number rearrangements that are very difficult. So for example, there's a known Portuguese mutation, Portuguese founder mutation, which is a transposable element, an ALU sequence that hops into the middle of a BRCA exon. It doesn't cause a copy number change, but it is quite pathogenic. Indeed, I know that Myriad's new MyRisk assay struggles on that variant because we've seen a negative patient report in a patient that we detected as positive and validated that. So some of these types of changes can be picked up by next-gen sequencing. Now, for example, large chromosomal inversions in place events, the type that are often looked at in pediatrics, were not considered in the scope of this particular assay. So we haven't looked at that particular question. Some we would detect, some we might not. Okay, maybe with that,